So an outline of what I'm going to talk about. I'll talk briefly about my research lab at Southern Illinois University, Carbondale, and the invasive species research that we do. And we do pretty much basic research, but I'll try to make the linkage from basic to applied research, applied being what perhaps many of you care about. How can we deal with this plant? And then I'll go through some of the different things we've been looking at in my lab to try and see if basic research can give us, identify perhaps some sort of Achilles heel for chaff flower. So I'll talk about size reproduction relationships. I'll talk about, does it matter if we control this plant? A bit about population growth, a bit about how variable it is, and this question about whether it's actually evolving as it's spreading, and a little bit about predicting its spread, where it might be going next, and then finishing up with some research gaps that we can identify. So in my lab, we, amongst other plant population and community ecology issues, we do look at invasive species. We've looked at several of them. We've looked at, as we'll talk about today, chaff flower. We've also looked at Japanese stillgrass, Microstygium verminium. Here's a picture I think that Chris showed earlier on. We've looked at Sericea, Lespedeza cuneata, and some work on some of these other species that are listed up here. So a variety of different invasive species, trying to understand the basic biology, really. And as I mentioned, we tend to focus on the left-hand side of this slide, the basic research, which we're just really trying to find out about the particular species. You know, what's their natural history, their population biology, how do they relate to the communities we find them in? And the hope is that by finding out some of this basic or empirical knowledge about these species, that can help us then develop practical applications, in this case, to try and control these species. So it is a continuum from basic to applied research, and so we tend to be on kind of this left-hand side of things. So to start off with the different topics, I'm going to talk about the size reproduction relationships. That the spoiler here is that the bigger plants produce more seed, and so these are some data from Katie Smith's master thesis, and each one of these different panels shows us the number of seeds produced in relation to different other parameters that are measured for these plants. So taller plants produce more seeds. The more inflorescences that have more seeds. The longer those inflorescences, Chris talked about how they elongate. You can see this in this picture here. The more seed, and the bigger plants, the more seed. So that's not really very good news, but you know, the bigger the plants that you see out there, the more seed they're producing, the more we have to worry about this spread that we've already been talking about. So bigger plants, more seed. So Chris talked about a little bit about where we find this plant, and experiments that we've done show that it does respond to richer soils. So on the left-hand side here, we see chaff flower above ground biomass in response to nitrogen addition. So it does better in richer soils. It also does better in sunny or open conditions, and this figure here shows that when you put it under shade, it doesn't do so well. So that kind of reinforces where we tend to find it in these edges of forests, sometimes doing really well. And so light gaps, forest edges, areas where there's a bit more light, the plant is going to be doing better, getting bigger, and obviously producing more seed, and also in areas where we have richer soils. This particular study compared chaff flower with some of the other plants in the same plant family. So we compared it with water hemp, Palmer amaranth, a rare plant in the amaranth family, bloodleaf, and also with soybean, obviously in a different family. So it's a perennial plant, as Chris mentioned. And one of the strengths of this plant is that it doesn't take much to get going and become perennial. And so some work that Katie Smith did was to grow plants and then to clip them back at certain stages. And so if you look at this plant on the left-hand side, Chris mentioned that it produces pairs of leaves at each node. And so as a young plant grows, you can see these pairs of leaves. And what Katie did was to clip the plant back at different nodal stages and see how long it took to regrow. And what she found, unfortunately, was that once it's got 3 nodes, and this plant here maybe has got what, 1, 2 nodes. Once it reaches that 3-node plant, it's going to come back. So it doesn't take much to establish, in other words, to become a plant that's strong enough that some sort of mechanical damage, that could include mechanical management. Once it gets 3 nodes inside, and that's a small plant, it's capable of regrowing. And she did a couple of trials. In one case, the 3-node plants took about 30 days to come back. And another trial, they only took about 5 days to come back. And you see the plants that had more nodes, of course, also came back as well. So it's a plant that regrows following damage pretty quickly and easily. It's only got to be 3 nodes in size. And if we do control it, and we're going to talk more about this in another session about methods of control, but as Chris mentioned, it's one that is fairly susceptible to herbicides, and so we do have good control methods for it. An important thing is, though, if we control it, does the native system come back? It's all very well controlling an infestation, but we also want the native biodiversity to come back. And in this study, the plant was controlled with a range of different herbicides, and then looked to see what the communities were like after controlling it. And this panel here on the left shows each one of these points is a experimental plot that was treated with one of these herbicides on the bottom left here, or not, which are the green circles. And in this sort of analysis, each one of these points, the closest they are together in this ordination analysis, the more similar they were in terms of community composition, in terms of the abundance of all the different species. And what we see is that the control plots were within this orange polygon here. There's one exception, but we're within this orange polygon, whereas the plots that were treated with the herbicide were outside of that mostly, and over here. So the message is that if you control it, then the community that grows back is going to be more variable and more different than the plots were beforehand, which is good news. We want the native plants to come back. Now, we don't know much about this. We don't know much about how general this sort of result is. It'd be useful to do this in other systems to see, how good is it when you really, how long does it take to restore the native communities? And this particular study was done, I think, down at Cypress Creek in southern Illinois, and the native community is not too diverse. If you look at the list of species down there, it also includes euonymus and some things like stinger nettle. So it's not a particularly exciting community, but nevertheless, it did come back following treatment. So that's the good news. We really need to know more about this. Now, in terms of how fast this plant grows and how fast the populations grow, we've done some studies looking at survivorship. And on the right-hand side, this figure shows that seedlings that germinate over the course of 250 days in a season, somewhere between 50 to 70% of those seedlings will survive. In other words, once those seedlings come up, over half of them are going to survive. And of those that survive, more than half of them are going to flower. So this plant really establishes well, is what this tells us. The seedlings that come up, most of them will survive, and most of them are going to flower. And of course, that's not what we want. It's a perennial plant, and on the left-hand side, this figure shows us that the survival probability is a function of height. Once a plant reaches about 20 centimeters in height, it's got 100% probability of surviving to the next year. So it doesn't, I've said already that once they get to 3 nodes, they're perennial. Once they get to 20 centimeters in height, they're going to be there next year as well. So this is a plant that once it establishes very easily and survives till the next year. So it's a pretty persistent plant once it reaches a site. Now, how fast do these populations grow? They grow fast. And we've seen that it's spreading. So here's some data that Lauren Schwartz did for part of her PhD dissertation, in which she counted seedlings, individuals, from one season to the next. And you can calculate then something called lambda. That's the equation on the right here. We simply compared the number of individuals in a location from 1 year and to the next year, when you divide one into the other. So you might have, in this case, 3,900 plants 1 year and 1,700 plants the year before. So you divide one to the other, you get figures 2.3. If the number of plants from 1 year to the next is the same, then this number will end up as being 1. So that's the benchmark. If 1 is a population that's not growing. So when Lauren did this in a couple of different years, she compared chaff flower populations with a couple of our bad agricultural weeds and Aracini rhizomatosa, which is a rare plant in Illinois, bloodleaf, that's in the same plant family. So these are all in the same plant family. And so these are the lambda values, the population growth rate values, comparing chaff flower with these other plants. And so we can see the chaff flower in both times that she looked at was values greater than 1, indicating growth. The amaranth weeds had positive population growth, as you might expect. 0.97 is pretty close to 1. But positive population growth, we expect that for bad agricultural weeds. And the rare plant, as unfortunately we might expect, had negative population growth. So these are just numbers. So I took these numbers and I put them into a population growth model to just give us an idea of what these really look like. So in these graphs, then I started with 10 individuals and I ran the model for 10 years. So on the left-hand side, we have the data for the 2 agricultural weeds. And we see, so for Palmer amaranth, starting with 10 individuals in 10 years, according to this lambda value of 1.22, we're going to end up with 75 individuals. For Amaranthus tuberculatus, starting with 10 individuals, we end up with 50 in 10 years for lambda 1.18. Here's chaff flower, lambda 1.79. Start with 10 individuals, goes up to nearly 3,500 individuals. Notice the y-axis is not scaled the same for all these 4 graphs, but you can see the exponential growth potential for chaff flower. So it really is a plant that can spread very, very fast, faster than 2 of our worst agricultural weeds. And here's the bloodleaf, the rare species we have with lambda 0.68, some 10 individuals, and this would be projected to be gone in 10 years. Now, these are exponential growth models, and of course, in nature, things don't grow exponentially forever. There's a carrying capacity, and things should level out or even decrease after a while. But this shows us the potential. In the absence of any controlling feature, either natural or management, chaff flower has the potential to grow faster than a couple of our worst agricultural weeds. Now, what leads to this fast growth? We can look at this in more detail, and I'm just going to touch on this very, very briefly. But it's basically the propensity of chaff flower plants to do well in terms of size. On the bottom panel, this shows us for Palmer amaranth, and this shows us a comparison of size at one time, the height versus the next time. And this is like a heat map of the importance of height to that population growth rate that I just showed you. And it shows for palmer amaranth, that tall plants are really driving that population growth. For chaff flower, it's showing that although the plants can get pretty tall, as Chris mentioned, population growth rate is really pushed, if you like, by or determined by these medium-sized plants. So again, it establishes easily, it becomes perennial easily, it's got high survivorship. And once it reaches this medium size, if you like, 50 to 100 centimeters in height, that's really going to determine the population growth rate. So once it gets a medium height, this plant is taking off, in other words. That's what this is telling us. Now we can put these sorts of studies together, or the data that went into the last few slides, which is based upon very careful monitoring of individual plants, tagging them in the field, going back time and time again, counting plants, looking at survivorship, collecting seed and so forth. And the sort of data that we have can be put together in a flow diagram, as we see on this left-hand side of this slide. And I don't really know where to start with this, but let's start with seedling emergence. About from our data, this is from Chestnut Hills, one of the sites in southern Illinois, of seeds that we know in the soil, about 36% of them were emerging. Of those seeds that emerged, we've already talked about this, very high survivorship, 86% survive to reproduce. And of those that survive to reproduce, 84% of them were found to be around the next year. Plants produce a lot of seed. I think Chris had this number on one of his slides, about 11,000 seeds per square meter of seeds are produced. But surprisingly, and we don't know too much about this, most of them don't end up on the soil surface. So when you put soil traps out and you look, how many seeds we find on the ground, we only get about 1% of those seeds that were produced. Nevertheless, of those seeds that do reach the soil surface and get into the soil, about 77% of them are viable. And as I said, 36% of those emerge. So there's some clear areas of loss to this plant. Most of the seeds that produced maybe are being dispersed off-site, high dispersal that we've already talked about. Maybe they're being eaten or they've got pathogens or something, we really don't know. But enough, it produces so many seed that enough are produced that it really can grow fast, as we've already seen. And as we've mentioned already a little bit, we really don't know too much about what happens in the soil. We don't know much about viability in the soil, or pretty much nothing. We don't know about dormancy of seeds in the soil, so forth. So this is an area we really need to know more about. Now, I asked the question at the beginning, or in my title, about, is this plant evolving? And this is something that investigations of other invasive species have found, that as they spread through the range, they tend to evolve, become adapted to the local conditions. Is chaff flower doing this? So what Travis Neal did in my lab was to look at plants from across the range in the United States and compare them to plants from Japan. And he was able to do that by going online, and you can actually see digitized herbarium specimens from Japan. And this is fantastic. You couldn't do this about 10 years ago, but now you can. So here's one specimen, and it's got collection information, it's got a scale on it there. So he was able to make a load of different measurements. So here's the root, and so you can measure the height of the plant. You can measure the number of inflorescences, the size of them, the size of the leaves. Looks like got something's been eaten them, and make a load of different measurements. And he collected plants from some populations in our region, including Warfield, the original population, the founding population, and some other populations across the dispersal range, and made similar measurements. And so then he looked at these plants. This is a principal components analysis, which is a method of looking at all the measurements together. And again, points each one of these points is a single individual that was measured, and the points that are closest to each other are those that are most similar in terms of all the things he measured. So look at this, and we can see the Warfield site, the yellow circles. The plants from Asia, or Japan specifically, are the gray circles. And look where they are. And then look at the points from the other populations, and they're all in this restricted area. So what this suggests is that the plants in Japan are quite variable. The plants growing at Warfield are as variable, almost as variable as the Japanese plants, but the plants that are spreading across our range are less variable. So this kind of reinforces the idea that Warfield is the founding site, and perhaps dispersing from there a subset of those plants are what's dispersing across our range. A subset of aggressive plants, but a subset of the total. Now, as to the question, is it evolving as it spreads? There's a couple of ways to do this. One way is to do some genetic analysis, and we have not done that. Another way is to take plants from different populations and grow them together and see how different they might be. So Travis did that. He did it in the greenhouse and also at one of the sites, the infestation sites. So he took plants from different populations and grew them in soils from different sites. So here we have pots with different soils, and you can see some different colored soils there, darker one and a lighter one there. And each square here would be a soil type and a source population. And some were kept in the greenhouse, others were put out, pots were sunk into the ground at Chestnut Hills. So we had 6 seed sources and 3 soil sources. So what was found? Well, soil makes a difference. So here's the height of plants from grown in 3 different soil types, from Warfield soil collected from Warfield, that founding site, from Henderson, Kentucky, and from Chestnut Hills in southern Illinois. And we can see that the tallest plants were from Warfield, but on the Warfield soil, sorry. So this is irrespective of seed source. So plants growing in the Warfield soils grew tallest, plants growing in the Henderson soils grew shortest, and Chestnut Hills in the middle. There were not really clear differences in the soils between these sites. The Warfield soils had higher phosphorus and higher calcium and higher pH. The Chestnut soils had higher potassium and higher nitrate levels, and the Henderson soils had higher magnesium levels. So, but soil seemed to make a difference. In terms of source population, if we were finding that the plants were evolving to the local conditions, we would find what's called a home site advantage. In other words, plants grown in their own soil would do better. We didn't find that. So on this panel here, the first set of bars is the biomass of plants in Henderson soils, and each separate bar is one of the source populations. And we should expect to find that the Henderson source plants are doing best in the Henderson soil. They weren't. We would expect to see Chestnut Hills plants doing best in chestnut soils. That wasn't the case. And we'd expect to find Warfield plants doing best in Warfield soils. So there is an interaction between soil and source population here, but it's not clear really what it means. But it shows that there's some differences amongst source populations. It seems to be related to soil, but it's not a clear, certainly not very clear evidence that the plant is evolving to the soil types that it's finding across the range. But something's going on. We'd need to know more about that. And the final study I want to talk to you about is this issue of predicting spread. Where's the plant going to go in the future? So Xian, we did a modeling study of a bunch of invasive species. And what she did was to use a computer model, which takes known locations of, in this case, chaff flower, that's the green dots in this map here, for our Southern Illinois region. All these are known locations. And then from each known location, work out the environmental data associated with that point, and then use the computer model to predict, based upon those same environmental data, where the plant is predicted to be able to grow. And so here we see the documented locations, and in the kind of the heat map colors here with the, it's a probability map. So the lighter the color or the red of the color, the higher the probability that the plant could grow in those locations. So we see that chaff flower is filling in some of the gaps between the known locations. And the three environmental factors that were most related to these projections are, first of all, soil type. And there's this bottom left-hand bar graph here. And it's particularly well suited, it seems, or likely to spread when it finds itself on these, on the left-hand side, these central Mississippi Valley wooded slopes, or this number 6 here, which is Kentucky and Indiana sandstone and shale soils, or number 8, which is a southern Mississippi Valley loess soil. Second important environmental factor related to this predicted spread is distance to river. We've talked before about how it seems to be spreading down the river systems. And so the closer to a river system it is, the higher the probability of it being able to establish. And thirdly is temperature, minimum temperature. This is in centigrade, so a minimum temperature of about 7.5 degrees is optimum for the chaff flower. Now, the fact that temperature is important suggests that perhaps we might see some further spread under climate change. And so Xian then took these models and then added to them climate change projections to see where chaff flower could go with climate change. And so here are outcomes of that modeling. And what she did was to compare some of the different climate projection models. And so with different levels of CO2 concentrations. And so the worst of these models is called the RCP 8.5. It shows an even more dramatic increase in CO2 than we've been experiencing so far. Another one was the so-called Paris Agreement goals, which is the RCP 4.5, which is this purple colored one on this bottom right-hand slide here. She also compared the other two. But just to summarize findings, basically what's going to happen is that chaff flower is going to fill in the gaps. So it's projected to be more abundant than it was with the current scenario, current projections that I showed you in the last slide. And this is going to happen quickly. These panels from left to right show 2021 to 2045, 2046 to 2070, 2071 to 2095. And we can see that regardless of scenario, 4.5 or 8.5, it's going to spread dramatically, it's projected to spread dramatically by 2045. So climate change is just going to allow chaff flower to spread even more, is the outcome of this modeling. Now, what do we do with our research to try and make it get out there so people know about it? There is a fact sheet on the ecology and control of Japanese chaff flower that we contributed to, that's available on the River to River website. Nick can tell us more about that, its availability, but some of the data from our studies contributed to that. Of course, we made presentations, and his Travis, when his hair was a bit shorter than it is today, making a presentation, and we publish our stuff in the academic literature. And if you are interested in any of publications, I'll be happy to send you lists or send you copies. Now, so what's next in terms of what we need to do about research? So this figure is one that you maybe have seen a version of this before, that shows you've seen a version about the spread of invasive species through time. In this particular version from Garcia Diaz paper in 2020, they showed the spread of invasive species through time as a function of the knowledge about its impacts, because after all, it's not just does it occur, but is it making a difference to our natural areas that's relevant. And what they propose is that early on, when a species is just invading, we need to prioritize early control. And that's when we need to know about basic life history, what's happening to it, the seeds being produced, and how long they're living and how long they're surviving and so forth. And as an invasive species spreads more, we need to be more concerned with what they call impact-based management, the effects of the species. And so initially we were at the point with chaff flower that very basic life history research, natural history, we need to know about the plant. Compared with, say, Lespedeza cuneata sericea, where it's been around for a long time, and we really need to be concerned with what it's doing to the environment. I think this is where we started with chaff flower. We're at this point now where we know there's still more we need to know about its basic life history, but we need to know more about its impact, what it's doing to the environment. So there are gaps in our research, and these are just some of them. As I mentioned, we don't really know what happens to the seeds in the soil. We really don't know how long they live in the soil. I assume it's several years. We don't know too much about what chaff flower is doing to the soil or to the microbiome. That's the bacteria and fungi associated with the root systems. We did do some work looking for mycorrhizae. Many plants have fungi associated with the roots, and we saw some evidence that perhaps there might be mycorrhizae in the soil, but we weren't for sure. We did find, Leah Matchke found that if you added fungal inoculant to sterile soil, the plants do better, suggesting they maybe benefit from mycorrhizae. Mycorrhizae can be an issue in plants. Garlic mustard is one that through allelopathy destroys the mycorrhizae of other species, so it can be a relevant factor. Someone mentioned allelopathy early on, and again, we're not really sure about allelopathy. We've got some evidence that it produces some chemicals, although when Anna Fraley did a study, she took leaf extracts and found they basically made other plants grow better, like a fertilizer effect. And so we don't know too much about that. As I mentioned before, we really don't know much about genetic evolution. We've got some evidence that perhaps something's going on, but we really need to do some genetic studies to look at that in more detail. We've talked several times already about dispersal vectors, and as mentioned by Nick, Travis was looking at deer fur, and this is one of his photographs showing some deer fur that have been pulled through chaff flower populations, showing the seeds collected. We don't really know about what I'm calling edge of range populations. Are the plants on the periphery, are they more aggressive, or what's happening there? We don't know much about them. And as I mentioned at the beginning of my talk, we don't know yet much about biodiversity recovery. In other words, if we do control it, do our natural systems respond and come back? How resilient are they? How long can they withstand a chaff flower infestation before they're really depleted? We don't know much about that. And so there's a lot of things we do know, and a lot of things we don't know. So I just want to thank the students in my lab that have been working hard on different aspects of chaff flower. I think I've got a picture of everyone in here, but if I've missed you out, I'm sorry. And so over the years, we've done quite a bit of work on chaff flower, but as I mentioned, there's quite a bit more we should do. I just want to mention, here's Nick putting his deer exposures up that he talked about, and there's Chris providing some help holding up that stake. But with that, thank you very much.